Welcome to this lecture on the poison apparatus in snakes. Imagine a doctor giving you a shot with a hypodermic needle. The doctor pushes the plunger, forcing the liquid medicine out of the barrel, through the hollow metal needle, and into your body. A venomous snake uses the exact same brilliant mechanical system, but it evolved millions of years before human medicine. Today, we are going to explore this incredible biological machinery, understanding both its anatomy and the fascinating physics that make it work. Now that we have introduced the topic, let us look closer at this hypodermic needle analogy. Why does this matter? Because understanding this mechanism explains how a relatively small, limbless animal can take down prey much larger than itself without having to wrestle or overpower it physically. In the snake's mouth, a specialized muscle acts as the plunger. A venom gland acts as the barrel holding the liquid, and a highly modified hollow tooth acts as the needle. When the snake strikes, it does not just bite, it actively injects a complex chemical cocktail into its prey. Building on that analogy, let us examine the actual biological components, starting with the glands and ducts. The poison apparatus is a highly specialized anatomical system. The venom glands are actually modified labial or salivary glands located on each side of the snake's head, just behind the eyes. These glands are crucial because they synthesize and store the venom, which is a complex mixture of proteins, enzymes, and toxins. From there, a narrow tube called the venom duct connects each gland to the base of a specialized tooth known as a fang, creating a closed delivery pathway. Following the venom's path from the duct, we reach the delivery mechanism itself. Fangs are enlarged, sharp maxillary teeth that are either deeply grooved or completely hollow, functioning exactly like that hypodermic needle we discussed. But a needle needs a plunger. Surrounding the venom gland is a powerful muscle called the compressor glandulae. During a strike, this muscle forcefully contracts to squeeze the venom out of the gland, through the duct, down the hollow fang, and into the victim's tissue. This muscular action is what makes the injection so rapid and effective. Let us visualize everything we just discussed with this anatomical diagram. Here, you are looking at a cross-section of a snake's head. Notice the large, light blue oval structure behind the eye? That is the venom gland, where the toxic cocktail is stored. Wrapped around it in red is the compressor glandulae muscle. When that muscle contracts, it forces the venom forward through the narrow venom duct, which you can see connecting to the base of the fang. The arrow shows the venom flow traveling right through the hollow center of that sharp, curved fang and out the tip. Having seen the anatomy, you might wonder why snakes evolved such a complex and energetically expensive system in the first place. The primary reason is predation and survival. Because snakes lack limbs to hold down struggling prey, venom allows them to strike quickly, release the prey, and wait for it to die, thereby avoiding injuries from sharp claws or teeth. Furthermore, snake venom is packed with digestive enzymes that begin breaking down the prey from the inside out, even before the snake swallows it whole. Interestingly, understanding this evolutionary marvel is also vital for human medicine, helping us design better antivenoms and blood pressure medications. Now that we understand the biological purpose, let us look at the physics of how this venom is actually delivered. To truly understand how efficiently a snake delivers venom, we can look at the physics of fluid flowing through a tube. Because the hollow fang acts as a microscopic pipe, The flow of venom is governed by strict fluid dynamics principles. Different physical factors, like the size of the fang and the thickness of the venom, directly affect how fast the venom is injected during a split-second strike. By applying a mathematical model, we can bridge the gap between biological anatomy and physical mechanics. This brings us to the mathematical model that governs this flow, known as the Hagen-Poiseuille equation. Conceptually, this formula calculates how much fluid can be pushed through a pipe under pressure. Let us break down the symbols. Q is the volumetric flow rate, meaning how much venom is injected per second. R stands for the inner radius of the fang, and L is its length. Delta P is the pressure difference generated by the snake's compressor muscles. Finally, the Greek letter eta represents the dynamic viscosity, or thickness, of the venom. Notice that the radius is raised to the fourth power. This means even a tiny increase in the fang's width massively increases the venom delivered. Conversely, a thicker venom with a high eta requires the snake to generate much higher pressure. Before we move on, let us see this formula in action with a concrete example. Imagine a rattlesnake striking its prey. We are given the inner radius of the fang as 0.0005 meters, the length as 0.01 meters, a muscle pressure of 20,000 pascals, and a venom viscosity of 0.005 pascal seconds. We want to find the flow rate, Q. We substitute these metric values directly into the Hagen-Poiseuille equation. First, we calculate the numerator by taking the fourth power of the tiny radius, multiplying by pi and the pressure. Then we calculate the denominator. Dividing the total driving force by the resistance gives us a final flow rate of roughly 9.8 times 10 to the negative 6th cubic meters per second. Physically, this means the snake injects nearly 1 milliliter of venom in just a tenth of a second. Now that we have successfully used the standard formula, let us consider a different scenario where we know the required flow rate but need to find the muscle pressure. To do this, we must derive a rearranged version of our equation. We start with the standard Hagen-Poiseuille equation. Our goal is to isolate delta P, which represents the pressure generated by the compressor glandulae muscle. Mathematically, our first step is to eliminate the fraction. We do this by multiplying both sides of the equation by the denominator, which contains the constant 8, the viscosity eta, and the length L. This brings all the resistance factors over to the left side with the flow rate Q, setting us up for the final isolation step. Continuing from our previous step, we now have the pressure, delta P, multiplied by pi and the radius to the fourth power. To completely isolate delta P, we simply divide both sides of the equation by those remaining terms. This gives us our final rearranged formula. This derivation is powerful because it reveals the intuition behind the physics. We can clearly see that the required muscle pressure is directly proportional to the flow rate, the venom's viscosity, and the fang's length. If any of those increase, the snake must squeeze harder. More importantly, it is inversely proportional to the fourth power of the radius, meaning that narrower fangs require exponentially more pressure from the snake's muscles to achieve the same venom flow. So, we have proven that this rearranged formula holds. This brings us to a real-world application involving a king cobra. The cobra needs to deliver a massive venom flow rate of 2.0 times 10 to the negative 5 cubic meters per second. We are given the fang length, radius, and venom viscosity. We need to calculate the pressure the compressor muscle must exert. We plug our given values into the formula we just derived. We calculate the numerator, representing the total resistance factors multiplied by the desired flow rate. Then we calculate the denominator, representing the geometric capacity of the fang. Dividing these gives a final pressure of approximately 18,863 pascals. This highlights the incredible muscular adaptations these reptiles have developed to push thick venom through narrow fangs at high speeds. We have covered a lot of ground today, so let us review the key takeaways. First, we learned that the snake's poison apparatus is a masterpiece of evolutionary engineering, functioning exactly like a biological hypodermic needle. We saw how the venom glands, ducts, and hollow fangs connect to form a highly efficient delivery system. We also discovered that the powerful compressor glandulae muscles provide the immense pressure required to inject the venom. Finally, by applying the Hagen-Poiseuille equation, we understood the fluid dynamics at play, specifically how the microscopic radius of the fang and the thickness of the venom dictate the physical effort the snake must exert. You now have a comprehensive understanding of both the biology and physics of snake venom delivery. Great job!